Hello everyone and welcome to the lecture for this video. So in this video, we're going to be going into the physiology of digestion and absorption. So we just finished talking about all the structures involved in the digestive system, and I hinted at all the enzymes and how you absorb these things and the different mechanical things and whatnot going on. So here we're going to be looking at that physiology in more detail, how we break down these macromolecules into their monomers. Remember, macromolecules are usually a chain of monomers outside of lipids, and we need to break all those bonds in between because we can only absorb the individual subunits. So how do we break all those down via enzymes, and then how do we absorb them? So we're looking at the physiology of digestion and absorption today. Now, one first part on this is mechanical digestion. So that's in the mouth. That's where that mostly takes place, and that's known as mastication. Remember, your masseter muscle is one of your main muscles of mastication. Then, in now to move all this stuff through your system, there's a wave called peristalsis. So peristalsis is the movement. So when we look at you know our GI tract, it's got those two layers. It's got a longitudinal layer of smooth muscle and then a circular layer of smooth muscle. Then inside would be the lumen where the food is. So imagine that longitudinal layer contracting. It would contract like this. That circular layer then contracts like this, squeezing like that. Now put those two together, you get this little wave that moves through. So that's what's occurring in the esophagus and throughout the canal. Remember, the stomach then has one more oblique layer, allowing for the churning and mixing in the stomach. But along with peristalsis, there's another event called segmentation. If you just squeeze the circular ones, you pinch. And that pinching, and this is common in the small intestine, that pinching is another form of mechanical breakdown of the food. And so that segmentation can also occur along with peristalsis in the intestines. Then, of course, I mentioned that churning in the stomach. So here, just an overview of the system. You have the mechanical digestion in the mouth. You also have a little bit of chemical digestion via enzymes, salivary amylase, and then lipase, so just carbohydrates and fats. Esophagus is just a tube that comes down. Then in the stomach, there's some mechanical digestion from the mixing in that oblique layer. Now there is also some chemical digestion of proteins and fats. There's some stomach lipase produced, but proteins with the production of proteases and pepsin in the stomach begin breaking down proteins. Then you can also absorb some lipids such as alcohol and aspirin in the stomach. Hence, if you ever have to, or someone that's gone to the hospital for drinking too much, they pump that stomach to prevent more absorption. And then in the small intestines here, you get all the releases from the pancreas and the gallbladder, so bile and enzymes. So you have mechanical digestion from segmentation that we just talked about, and then the chemical digestion of carbohydrates, fats, polypeptides, those are your proteins, and nucleic acids. All your macromolecules are finishing digestion here. And then you also absorb them here. So absorbers of peptides, now it's mostly all amino acids, glucose, fructose, those are all monosaccharides, so single units, fats, water, minerals, and vitamins. Now some things require carriers, so like vitamin B12 requires something known as intrinsic factor. Calcium absorption requires vitamin D. So any vitamin D deficiencies can lead to calcium deficiencies and bone density reductions. So little things like that. Some things do require other things to help carry them across the cells. And then your large intestine here, there is some mechanical digestion, but it's not as big of a deal at this point. There's no chemical digestion, so it's just your bacteria eating things now. And so you can absorb, ions, water, minerals, vitamins, organic molecules, but other than that, it's not a whole lot going on in the large intestine except for just being a bacterial warehouse. All right, so now let's go into a little bit more detail of chemical digestion. First macromolecule here is carbohydrate. So when you eat carbs, so sugars, they're usually in the form of starch or glycogen. Glycogen is how we kind of store glucose, so in our body. So first, in the mouth, you release salivary amylase. Salivary amylase can break things into polysaccharides and disaccharides. You don't have to remember the names of all these polysaccharides and disaccharides, but just as an example here, sucrose is typical sugar, like table sugar. Lactose, if you're lactose intolerant, you lack this enzyme here called lactase, and you can't produce the two monosaccharides from it, and then lactose ends up going through your system. And if you're lactose intolerant, you know the problems that lactose causes then, because this missing enzyme here called lactase. Sucrose is then broken down by sucrase, maltose by maltase, and all these are released by the pancreas. Now, they're released and then they're activated, and they're known as brush border enzymes for a lot of these. So brush border enzymes are enzymes that are embedded on that brush border on the microvilli on the enterocytes, which are the cells of the small intestine. But then here, these are the goals to break carbohydrates into monosaccharides, because that's how you can then absorb them. For proteins here, proteins, you have proteases. And now there are a whole bunch of different proteases that are released, like in your stomach, you begin to break down. So not only does hydrochloric acid help, but pepsin also begins to help. And then the pancreas, so that begins breaking down. Then you enter the duodenum, pancreas releases a whole bunch of different enzymes. Now, they're secreted in the inactive form and activated out here, because if those proteases were active in the pancreas, you would digest your own pancreas and chemically break down the pancreas. So there's a way they control the activation once they get out into the duodenum. And the goal here is to take these chains of amino acids and break them, so these peptides, and break them down into the individual amino acids that you can then absorb. So that's what the small intestine is then doing. So you're doing that final digestion via all those proteases released from the pancreas, and then final absorption takes place there. And then here, protein digestion enzymes are also secreted from the brush border, so the final breakdown helps activate them. And then you absorb them, they go through the liver, and then go to wherever they need to go. The liver then regulates where they go throughout the body, and you do lose some of them in feces as well. So those are the amino acids, or the proteins. Nucleases, nucleotides, you break them down. So your nucleic acids, and you absorb the subunits. We're not going to talk too much about those, but you are what you eat. You do eat DNA. You do then break them down into the nucleotides that you then absorb and make into your own DNA. But it matches your sequence, so it doesn't matter. Lipids here, those are our fats. These are broken down by lipases released throughout. There's salivary lipase. There's some released from the stomach and some released from the pancreas. But the big thing here is bile, which does emulsification. And what that does is it kind of makes these little, they're called micelles, that surround the lipids. And the lipids would be in the middle. And then they can absorb through and just go right through the lining of the intestine and get absorbed into lacteal. So now I hinted a little bit about absorption. So how does absorption work? So there are three different ways it can do it. There's passive. So passive is just where it goes from an area of high concentration to an area of low concentration. So it just passively moves that. So, but here in the small intestine, there's a barrier we have to go through. There's a cell. So lipids can go right through that barrier, so it's not a problem. Facilitated diffusion is where it has a transporter that facilitates that passive diffusion. And then active transport, this one is the one that requires ATP, which means it requires energy. Now, most stuff being absorbed requires this energy. So let's look at this little table I have of the different macromolecules, or not macromolecules, but monosaccharides or amino acids or fatty acids being absorbed in the alimentary canal. So here we have different carbohydrates. You see here, these are co-transported with sodium ions. So here, sodium becomes really important. And I'm actually going to draw this out. So if we have a cell here, it's got the microvilli up here. So this is the absorptive cell, an enterocyte. What happens is that you actually have a sodium-potassium pump in this cell. So what it does is it pumps sodium out and potassium in. And then so it actually pumps 3 sodium out and 2 potassium in. You don't need to worry about that. But what happens is you get a low concentration of sodium inside this cell. This requires ATP because it's pumping against its concentration gradient. So out here you have a carbohydrate, you also have sodium ions. And the way a lot of glucose, galactose, and amino acids here do this as well, they have its secondary active transport. So you have a co-transporter that when, say, glucose comes in, sodium comes in. And if sodium wasn't coming down as gradient, glucose wouldn't either. And then you get those in here, and then you can pump them through the other side, and then here's where they can be taken up into the blood. So long story short, without this ATP changing the sodium concentration inside the cell, this won't work, and you won't be able to absorb anything that says sodium in this table. So that is active transport requiring ATP. And then lipids here, these just go through regular diffusion into the lacteal. So don't worry about the different types of lipids, but they are diffusing right into the lacteals via these little structures called chylomicrons. So it's a protein lipid structure that help the absorption. And then also there is some short chain fatty acids and glycerol. Don't worry about all these ones too much, but some of these can go into the capillary blood and be absorbed in there. So the rest of these all do get absorbed into the blood as well. And then nucleic acids, which of course we didn't talk about much, that's also active transport, but it varies depending on which nucleic acid we're talking about, and that goes into the blood as well. Okay, so these are our macromolecules, but what about water? Water gets absorbed via osmosis. And throughout this process, we talked about all these glands releasing things. So when you drink water, your saliva releasing tons of water. Gastric secretions are aqueous, tons of water. Liver, the bile is water. Pancreas has tons of water in it. And then your intestines also have lots of secretions in it. So in the end, you have a very large 9,000 milliliters of water that eventually gets released. That's 9,000 milliliters is a lot. So you, of course, don't release 9,000 milliliters constantly. Here, you can see how you reabsorb 78. So when you begin absorbing the nutrients back in, so the amino acids, the carbohydrates, your water follows via osmosis or passive diffusion. So there are aquaporins in the channels as well. So as you move the nutrients in, the water follows. And then in your large intestine, you finish reabsorbing more of it. And then you do lose a little bit in feces, of course. So if you have diarrhea or something like that, that's why it's more watery. It's because it doesn't have as much time in your system to reabsorb. So you got to be careful of dehydration in that case. But this is all osmosis, which is the movement of water. But you do reabsorb. Most water occurs in the small intestine, but a little bit does occur in the large intestine as well. Now, all of this requires lots of control. So the last little thing we're going to talk about here are how 3 hormones, gastrin, secretin, and cholecystokinin, all control this. There is also some neural regulation. Think about your thought of food. You start producing saliva. So just your it's called your cephalic reflex. The thought of food, you start increasing gastric gland release in the stomach. So HCl, pepsin, and also saliva. If you ever heard of Pavlov's dog effect, that's what that's referring to. But here, that's neural regulation. You also have your gut brain. Your gut brain is your enteric nervous system that's responding to all the reflexes, and a lot of that is regulated via these hormones. So here, gastrin, secretin, and cholecystokinin, or also known as CCK, which is a little easier to say. So here, we're comparing gastrin, secretin, and CCK, and where it's produced. So produced by G cells in the stomach. Small intestine for secretin, and in the small intestine for CCK, where these are the organs they target. Gastrin targets the stomach and small intestine, pancreas and stomach, gallbladder, pancreas and stomach. But here is the important part. What causes the release? So it's some sort of reflex. So presence of food in the stomach causes gastrin. Gastrin causes gastric acid. That's where it gets its name. Gastrin, gastric acid, stomach. So this causes HCl secretion in the stomach. Now, when that food goes into the duodenum, or the small intestine, it's acidic. Remember, it's called chyme at that point. That acidic chyme, there are chemoreceptors in there that will be like, oh, hey, this is acidic, targets the pancreas to cause bicarbonate secretion. So secretes bicarbonate from the pancreas. Also inhibits gastric glands in the stomach as well. And then CCK, due to the presence of fatty acids, amino acids, and chyme, so saying mainly here fatty acids, we need bile. So it stimulates the gallbladder to produce bile and then also causes the pancreas to release enzymes to break down those proteins. So all of these are just hormones that are just part of the digestive system that play a very, very important role in it. But so this finishes the lecture portions of the digestive system. I hope you enjoyed all of the breakdown of food stuff and how you absorb it and how you get rid of it. So very, very important system in our body. A little bit easier to break down compared to the other systems, like the nervous system. But if you have any questions, like always, please reach out and let me know. But I hope you enjoyed this little lecture here going over the physiology of digestion and absorption, and I hope you learned something. Alrighty, see you all next time. Bye bye.